Thank you. Good morning. Thanks everyone for being here. I'm excited to share the results of this environmental DNA survey for the proposed federally endangered salamander mussel. The survey was completed in partnership with the Ohio Department of Transportation. So I'll start with a little background on the salamander mussel, which was proposed endangered in 2023. This mussel has a unique reproductive strategy in that it uses an aquatic salamander species called the mud puppy as a host for its glaucidia or larva. This is unusual because every other freshwater mussel species in North America uses a fish as a host for reproduction. Also probably explains where the name salamander mussel came from. So the salamander mussel and the mud puppy prefer similar habitat types, and those habitat types include rock crevices, boulder slabs, and overhanging banks. And because of that habitat preference and the small size of the mussel, it can be very difficult to find using traditional survey techniques. So the purpose of the study was to evaluate the use of eDNA as a tool for detecting salamander mussels at different densities, habitat types, flow regimes, and to compare that to the results of traditional sampling. In Ohio, the Blanchard River is proposed as critical habitat for the salamander mussel, but the only record of the salamander mussel in the Blanchard is one dead shell recorded in 2009. So another goal of this study was to try to determine whether the salamander mussel is present in the Blanchard and where it is present. Oops. Okay, so environmental DNA works like this. As mussels filter and release water, they're also releasing cellular materials into the environment. We can then collect and analyze water samples to determine what DNA is present in water. The concept works the same for other aquatic organisms, such as the mud puppy. So using eDNA is actually less intrusive than a traditional mussel survey because it doesn't require the physical collection of the organisms for identification. And it can also be less costly and more effective at detecting rare species than physical search methods. Our study included 5 sampling locations in areas of proposed critical habitat in Wisconsin, Kentucky, and Ohio. In each of these locations, we examined the water samples for both salamander mussel and mud puppy DNA, and also conducted visual tactile surveys so that we could compare the presence of eDNA and the density of the mussel population. So we started by sampling in sites of known occurrence, and at every one of our sites of known occurrence, we had positive eDNA detections for both salamander mussel and mud puppy. The numbers shown represent the number of salamander mussels collected from the sites. So we've had a pretty wide range from over 700 individuals collected in the Wisconsin River to just 2 fresh dead shells in Fish Creek. Suitable habitat in the survey areas also differed across the sites. So in the Wisconsin and the Licking, the habitat was sandstone slabs or rock shelves, whereas in Fish Creek, habitat consisted of undercut banks with overhanging vegetation. And that was the same type of habitat that was present in the Blanchard River as well. Okay, so now we can review our eDNA results in the Blanchard River. So this map shows all of our sampling sites for both eDNA, which is represented by the circles on the map, green being positive detections, and visual tactile surveys, which are represented by the stars on the map. Along the entire reach, we collected positive samples for mud puppy in 2 locations on the Blanchard. Here we go. And then we can compare our results for the salamander mussel. So we had one sampling site, which was positive for salamander mussel eDNA at the very downstream end of the critical habitat reach. We did not find any live individuals or fresh dead shells of salamander mussels in the Blanchard as part of our visual surveys. So in our sites of known occurrence, we had positive detections of both salamander mussel and mud puppy DNA, even when there were very low population densities. in the Blanchard, the detection of the mud puppy host and the detection of salamander mussel seem to suggest that the species is likely present, but in very low quantities. The species may not have been in the physical sampling area. It may have been very rare, or, as the detection was at the very downstream end of the reach, it is possible that the DNA was transported from somewhere upstream. So a few implications of the findings of this study. One is that we can use this information to better understand salamander mussel distribution, particularly within areas proposed for designation as critical habitat, such as the Blanchard River in Ohio. Another thing is that eDNA successfully detected two very different species types. Finally, our findings support the use of eDNA as a tool for determining the presence of cryptic species that may work in addition to or in lieu of traditional survey methods. And with that, I'll take questions. It depends on the system, and actually I did not have time to go into it, but we also did a transport study and found that the DNA tended to drop off after about, I believe it was 500 meters, but it totally depends on the flow, the time of year, and I think the studies have shown that it can be transported up to 7 kilometers. Is that right? Yes.
Right, I was just wondering, it's the last one of the question. Except that we propose the critical habitat, other implications related to that considered anyone that's critical habitat, or is that kind of beyond the study affected by your result?
So the results of this study, so the question was, did the results of this study, could they possibly affect the proposed designation of critical habitat on the Blanchard? I think that because of the positive detection of the salamander mussel, it's unlikely. Additionally, there's the possibility for critical habitat to be unoccupied. So I don't see that this study will have any effect on the proposed designation.
So, I don't know, how many samples did you do per site? Did you do a sample of the individual samples per site?
The question was how many samples were collected per site. I think the answer is that the sites varied, but several samples were collected at every site. Probably at least the samples were collected by transect, and I believe there were 4 transects-ish per site, and those were paired with cell searches for visual tactile.
And did you pull those almost per site?
So the question was, did we pull all of the samples and analyze them as one sample in the lab? And the answer is that we analyzed several samples in the lab so that we could have some repeatability.
Trying to find out if I know it's the top twice in the short. So if you we reach out to you, we give us a detail how you guys do it, if there's any machines or incubators or anything that tools that help you to do that and share with us. Thank you.
Yes, thank you. So for further questions, I have included Nate Marshall's information here as well. He was the lead of this study, and so he'd be happy to answer more detailed technical questions after the session.
Here. And so most of you use presence absence and most of the abundance.
The question is, did we do presence, absence, or abundance? And the answer is that we collected multiple samples at each site. And so we did have an abundance component that I didn't really have time to discuss during this presentation.
Here's about the visual surveys and elements. Would you be able to tell from if there's actual life in muscles or if it like you have this functional system?
The question was, without the presence and confirmation of live individuals with visual tactile surveys, is it possible to tell whether there actually are live individuals there or if the detection is related to dead shell remnants? I believe the answer is that the detection of the eDNA is likely associated with live individuals, but I think that's correct. So yes.